Okay, everyone, another scintillating lecture in the biological sciences. I think scintillating is one of the least utilized words in the English language. Second behind brouhaha, one of my favorite words. It means to have a big, just a lot of drama. A brouhaha occurred. There's a lot of drama, a lot of much ado about nothing. Very good word. Suggest you use it at some point in your life in casual conversation. But now we're on to molecular genetics. And the thing about genetics, as you probably saw with the classic genetics lecture, is you really got to be doing these things. And if your plan is to sit back idly like this and just soak in the information like a sponge, and this may not be the exam that you'll be, um, this might be the exam you're dropping because this is all because these genetics issues, they're all about doing. And it's very important that you write down exactly what I'm writing, draw whatever I'm attempting to draw. I draw DNA decently well. And and yeah, this is all about doing, writing, reinforcing, because if you don't do that, then what's going to happen is when you get to the exam, you won't be able to properly retain the information in order to be as successful as we both want you to be. You're probably seeing with this course by now that yeah, this is this is a fairly difficult course. It's biology, and it really what I tell my students is it doesn't matter if it's CP, honors biology, AP, biology is biology. You can't change the concepts. You can make them a little bit less complicated, but DNA doesn't become magically easier to understand because it's intro bio and not majors bio. So this still requires a lot of work. And what I'm hoping is that as you go through this course, you gain an appreciation at just how not only complex life is, but also how amazing it is that we can do things like sequence DNA, figure out the structure of a single nucleotide. So things like that is what makes biology such a fascinating concept. Now we're not always going to be on stuff you can't see, because sooner or later we're going to end up with ecology, which is one of the more students seem to like that one a little bit more because it's a lot more tangible. But enough talking on my part. We need to learn about some DNA. So let's go. All right. So DNA stands for deoxyribonucleic acid. And according to your chapter, it doesn't look like we're going to learn about RNA yet, but DNA stands for deoxyribonucleic acid. The main sugar base is called deoxyribose. And the nucleic part refers to the fact that it is in the nucleus. And DNA is a molecule. It is a molecule comprised of building blocks that you've already learned of monomers called nucleotides. Um, the number of nucleotides that make up a genome is the entire genetic information of an organism, can be anywhere from a couple million, if, well, actually, yeah, a couple million if you're a bacterium like E. coli, to 4.4 billion if you are a human. Viruses, which are technically not life forms, they have a couple thousand nucleotides. I know that the SARS-2 virus is something like, I think it's like 28,000 or something, 28,000 nucleotides. Fun fact, if you were to take the SARS-CoV-2 virus. All the SARS-2 virus in the history of its existence, so a year, that has ever been in anyone's body. So the total number of SARS-2 viruses in the history of this pandemic, it would probably only fill up about a teaspoon. That's how small viruses are. So anyways, a nucleotide is a building block of DNA, and it's based on a 5-sided sugar called deoxyribose. I'm not the best at drawing 5-sided sugars, but I think I did a decent job here. So this is a 5-sided sugar called deoxyribose. Deoxyribose. Now it's going to have a couple things attached to it. For starters, it's going to have what's called a nitrogenous base, which I'll go ahead and draw like this. It looks quite a bit different in as far as the molecular structure goes, but this will just give you an idea. So this is called a nitrogenous base. There's actually 4 versions of them. And it's going to be attached to a phosphate group, which if you remember from a couple lectures ago, a phosphate group is a phosphorus that's attached to a bunch of oxygens. Now, there's actually 4 variants of these nitrogenous bases. 4 variants, meaning that each nucleotide can have one of these 4 base variants. And one of them is called adenine, which we abbreviate with just A. The next one is called guanine, which we abbreviate with G. The next one is called thymine, which we abbreviate with T. And the last one is going to be cytosine, which we abbreviate with just C. Now, what is the significance of these bases? Well, the order of these bases in the DNA molecule serves as instructions to make proteins. It's basically a language. It is a series of instructions. So to give you a completely fictitious analogy, and one thing to keep in mind is that when we write the DNA code, we just write the first letter of the base. So let's say that we have a DNA molecule that is 5 nucleotides long, and it has this structure. And we'll talk about why it's two molecules or 2 strands in a second. So let's say that we have a DNA molecule that is 5 nucleotides long. This could be, if we're going to give the analogy of a cookie recipe, this could mean add flour. This could be add sugar. I don't know how to make cookies. I microwave everything. In fact, I'm trying to talk to Moorpark about teaching a class on how to microwave everything. Not true, but if I did teach that class, you'd be microwaving everything from filet mignon to hot cereal. All right, add sugar. And this could be add chocolates. I don't know. I'm just bake at 350. Serve on platter. I don't know. So this is a language. It is a set of instructions. In case you don't like my analogy of just cooking things, there is a website that every geneticist across the world uses. It's called the NCBI database. Let me go ahead and pull this up for you all. Very interesting stuff, the NCBI database. And it gets kind of laggy at times because, as I said, every geneticist in the world is using this. And when you have a point in the day when all the major countries are using this website, it can get laggy. So anyways, let's take the protein hemoglobin. And hemoglobin, which is abbreviated HBB, is a protein that is made that is in your red blood cells that grabs on to oxygen. Let's go ahead and look at the gene here. All right, this website is purposely non-user friendly because they don't want people that shouldn't be on the website mucking around. I mean, this is only for people who are very serious about genetics research. So you're not going to see any ads or anything or any kind of pleasant pictures. You're just going to be seeing a bunch of text. Okay. So let's find a nice reference to click on. You can even see mutations in order to. You can see where in the DNA code mutations are, which can cause certain disorders. So yeah, it's very interesting stuff. All right, so go ahead and look at this here. All right, just give me what here we go. All right, so this is the section on chromosome 11, which is where we can find HBB. And if you look here, this is the order of adenines, cytosines, thymines, and guanines that gives instructions for how to make the protein hemoglobin. And that's basically what our DNA code is, just a bunch of repeating A, T, C's, and G's. That tells our cellular machinery, which we'll be learning about probably next chapter or the chapter after. That tells our cellular machinery how to make proteins. So this contains the instructions for making hemoglobin. In case you're curious about the SARS-2 virus, you have scientists all across the world that have been sequencing this virus, trying to make a vaccine. Well, there's other reasons too. But if you look here, this has been sequenced something like, thousands of times. And if you look here, well, there's also been 103,062 research articles. So as you can tell, scientists have been pretty busy. So we can actually look here, and I don't really have time to muck around this, but I know that they have a section on here that has every single SARS-2 variant that has been identified. But as I said, is they purposely make this pretty hard to scan. So let's look here. All right, well, I'm able to find the Wuhan strain here, the first strain of SARS-2 identified. And this is the DNA sequence of the entire virus. Let's go down to the bottom. So this combination of A's, T's, C's, and G's is responsible for our global pandemic. I was close. There's 29,881 nucleotides that make up the genome. And what's amazing about this is that the Pfizer and Moderna vaccines, they've identified the part of this genome that codes for what's called the spike protein. And what they've done is that they've copied just the code for making the spike protein, and they've isolated it, made it into a vaccine. So what happens is that this vaccine has your cells, your own cells, make just the spike protein because they have the instructions for making it. The spike protein is harmless, so your immune system will respond to it. Start making spike proteins. The immune system will respond to it, make antibodies, and if it ever sees the SARS-2 virus, then it will identify those spike proteins and neutralize it quickly. So it's very amazing technology. All right, so back to the whiteboard. Now, our genome is based on these nucleotides. So the most important part is going to be the nitrogenous bases, but there's much more to the molecule than that. So I'm going to erase this. Now, this is a double-stranded molecule, which you'll find out why as we get into DNA replication. Erase this. What's this? And this. This has got to go too. Let me go ahead. I think I'm getting the hang of this typing-based stuff. I am kind of. So anyways, this is a double-stranded molecule. And so let me go ahead and draw the other strand here. And you're going to notice something interesting as I draw the other strands. You're going to see that it's actually upside down. It's actually running the other way. And that's because DNA has a quality called anti-parallelism. So these two nitrogenous bases, the two strands are connected at the nitrogenous bases. They're held together by weak bonds called hydrogen bonds. And I compare them to magnets because they're easily separated but also reformed. So these are hydrogen bonds that weakly hold together the two strands at the bases. And we call this characteristic anti-parallel. So the two strands run anti-parallel. We're going to write this. We're not going to. I'll just write it here. It might have to go away though. Anti-parallel. Think of it like the 101 north and south. They're both roads. They both go to the same places, but they just run in opposite directions. So the two strands run in an anti-parallel nature. They run in different directions. Now, these nucleotides, they're actually connected at this phosphate group. It's called a phosphodiester bond. And it's going to look like this. This is not a 100% accurate representation, but it's pretty close. And nitrogenous base. can go ahead and do another one here. Just like so. So I think you're starting to get the idea here. So we have nitrogenous base. And the same thing occurs here. I'm not going to go ahead and draw it exactly again because, well, we have a time frame here. And I don't think you guys want me to just be drawing nucleotides for an hour. All right, yeah, not perfect, but you get the idea. These bonds here are called phosphodiester bonds. They're very strong bonds. It takes a lot to break them apart. One of the things that can is radiation. So these are covalent bonds that hold together nucleotides. So they basically hold together the bonded DNA. And they really do not like to be messed with by radiation because they are going to scramble the DNA molecule, and the cell will no longer have a working copy of the DNA. That's a problem in cells that are constantly dividing because they'll forget how to divide properly. All right, so now we're going to talk about base pairing rules. And base pairing rules, what this means is that, and this is a very important concept, so definitely make sure you understand it. So base pairing rules. Based on the structure of the nitrogenous bases, adenine will always bind with thymine, and vice versa. Guanine will always bind with cytosine, and vice versa. So this means that in a genome, the number of A's, T's of AT pairs is the same. So what this means is that if there are 40 A's, there's going to be 40 T's. And the same thing on the other end too. If there are 50 C's, then there will be 50 G's. I'm not consistent here. I'll just do, here we go. What this also means is that, and this is very important, base pairing rules. So base pairing rules allows for the formation of a new strand using an existing strand as a template. So let's go ahead and practice this. If I were to say that one strand of DNA, that I'll call the sense strand. Oh, and we'll talk more about sense and antisense strands later. But one strand for DNA is called the sense strand. Let's say it's ATGCCA. If I were to ask you, based on this, to make the opposing strand using base pairing rules, which we call antisense, could you do it? And the question is, of course you can, because we know that whenever there's an adenine, there's going to be a thymine. Wherever there's a thymine, there's going to be an adenine. Wherever there's a guanine, there's going to be a cytosine. Wherever there's a cytosine, there's going to be a guanine, guanine, thymine. So this characteristic allows us to build a new strand using an existing one as the template. And in case you're curious, sense strand has the proper information to make a protein. The antisense strand does not. But you'll see why when we get to DNA transcription translation, why having two strands is necessary. Also, DNA is entirely contained in the nucleus. I don't think I added that. That's because you don't want your DNA molecule to be corrupted. So think of the nucleus as a safe and the DNA as your retirement. You don't want anything to damage it, which is why it's found in the nucleus. All right, and as we talked about before, and I don't want to rehash this too much, is chromosomes are segments of DNA. Recall that our DNA is going to be 4.4 billion nucleotides. So they are segments of DNA, which we've already learned about chromosomes ad nauseum. They're segments of DNA containing specific genetic information. Actually, specific genes. Now, genetic disorders, we talked about dominant and recessive alleles last class. But genetic disorders occur when genes have mutations that prevent a functional protein from being made. Protein from being made. Let's say that you're doing a chocolate chip cookie recipe and somebody made a mistake, and instead of a cup of sugar, they say a cup of salt. Yeah, you're still going to make something, but it's not going to be a chocolate chip cookie. Just one step wrong is going to cause the entire product to be corrupted. So actually, it happens very. I mean, it looks like a minor difference, but it's actually huge. So this could be the sense strand to make hemoglobin, for example. So this is hemoglobin. And something could happen because when DNA is copied, a process we'll talk about next, is sometimes there's an error. Now, animal cells, we have the ability to fix the error, but things like viruses don't, which is why they mutate so fast. So what can happen is that there could be a random mutation that changes this E to a T. So instead, the new code will be this. Everything is the same except this one nucleotide. So this is going to be dysfunctional hemoglobin. Just one nucleotide change. The disorder sickle cell anemia is caused by the same thing, a single nucleotide change. Now, once in a while, mutations actually cause a protein to be more functional. Or other times, it's going to create a new allele, a new variant. So, for example, a small mutation can change the gene for hair pigment from brunette to blonde. But many times, it just causes the protein to lose its functionality either fully or partially. All right, now when we come back, we're going to talk about DNA um, uh, replication. Very interesting, very interesting concept. All right, see you for part two. Well, it's time for our part two of our molecular genetics lecture. And we're going to talk about DNA replication. So, just to jog our memories here, well, specifically your memory, in the cell cycle, there is the S phase, which I'm sure you remember quite fondly. And during the S phase, the purpose is to prepare for mitosis. So, if you remember here, the cell cycle, and of course, the biggest thing in the cell cycle, the one that we're going to focus on right now, is going to be the S phase, which, if you remember, is the part where DNA is replicated. Replicated, well, I mean, means copied. Let's just call a spade here. All right, now we see here that the purpose of mitosis is you're taking one cell, right, and you are making two identical daughter cells. So what happens in the S phase, if we're going to do a not super realistic image here, the purpose of the S phase is you are, let me just make sure I'm going with the outline here, is you have started off with one DNA molecule. DNA is a double helix that's kind of shaped like this, so to speak. And it is going to become two. And you're probably going to be looking here and saying, wait a second, why are you keeping one strand red and the other strand a different color? And you'll see why when we get to the actual process. But during the S phase, one DNA molecule is going to be replicated and turned into two. So if that's the case, what happens is that prior to mitosis, when a cell is finished with G2, it's basically, and this is only a half truth here, but I'm just trying to illustrate a concept, is you're going to go from two DNA molecules in the pre-mitotic cell, and then in the post-mitotic cell, each one of them has a DNA molecule. So that is the purpose of DNA replication. To give you an example, in your skin is actually, and I'm sure I've said this before, your skin's actually quite dead, at least the part that you can see. And your skin is constantly flaking off. We call that dandruff if you find it in your hair. But yeah, your skin is constantly flaking off, and it's being replaced by new skin. Why is it that you can make new skin? Because you have stem cells. So stem cells are cells that can become any other cells. You have stem cells that are constantly undergoing mitosis. So I'll just say that this is a skin stem cell. And it is constantly going to replicate and make new daughter skin stem cells. Well, I'll just say skin cells because they eventually differentiate into what's called a keratinocyte. But that's basically the idea behind this. Is the idea behind DNA replication is you are going to be multiplying cells, and because of that, each one needs its own copy of DNA. Now, we have an interesting thing with DNA replication. Interestingly enough, the DNA molecule cannot just clone itself. And they actually thought that DNA, well, there were three theories with DNA replication. So three theories with DNA replication. And just to note here, because we're going to be talking about a couple of ways that genetic code is duplicated. DNA replication is an exact copy of the DNA molecule in one cell. So that exact molecule is being copied. Think of it like think of it like a copy machine. I guess that's an easy way to put it. So there are three major theories. One was called conservative. The other one is called semi-conservative. And the third one is called dispersive. Now, the idea behind conservative is that the DNA molecule just basically cloned itself. So something happened through some process, and the DNA molecule just clones itself. So that was the conservative. It just copied itself. They found out that was not the case. Dispersive, interestingly enough, what they thought is that, and this is a super brief summary, but what they thought is that the DNA molecule kind of shattered itself, and each piece developed new genetic material. So it's kind of like scattered, almost like taking cards and throwing them on the ground and then picking them back up. So with the dispersive, basically, what they thought is that there were fragments, and each fragment kind of replicated new genetic material. We also know that dispersive ended up being not quite right. I think the idea behind it was semi-sound, and you'll see why. But semi-conservative turned out to be the right one, and basically, what the idea was is that the DNA molecule actually separated into two strands, and each of these strands developed a newly synthesized strand. So the semi-conservative model actually ended up being the correct one. Semi means kind of, conservative means stays the same. They are not wrong, it kind of stays the same. So with the semi-conservative model, one strand separates and makes two separate strands. So we're going to go through the steps here of DNA replication using the semi-conservative model. Using the semi-conservative model. Conservative model. Okay. So I'm going to draw a DNA molecule here. Recall that it's double-stranded, and I'm going to write down just the bases. So this is the code of each DNA strand. And recall that it's connected using hydrogen bonds. And as talked about earlier, is the base pairing rules. Wherever there's a T, there's an A. Wherever there's a C, there's a G, and vice versa. So this is our like original DNA molecule. Now what's going to happen is the first step is an enzyme, a catabolic enzyme called helicase, which I'll go ahead and put down here, helicase. It is an enzyme that breaks hydrogen bonds, between the two strands. All right. So what this enzyme is going to do, and this is step one, the enzyme helicase is going to run through the middle of this DNA strand. And as it does so, it's going to break apart the hydrogen bonds, and it's going to separate these two strands, like so. So we see now that they are separated. And that's because helicase, the enzyme, it's been compared, and it's a pretty good comparison. Oh, oops. Of a zipper unzipping a jacket. That's basically what helicase does. Is it breaks apart the hydrogen bonds, causing the two strands to separate. Now, next is an enzyme, a very important enzyme called DNA polymerase 3. Yes, in case you're curious, there's two other ones. If you want to take majors bio, we'll talk about that ad nauseum. But DNA polymerase 3 is the main one, because what it does is following base pairing rules, it reads one strand and synthesizes a new one. So what it's doing is it is going to build a new strand using the existing strand as a template. So following base pairing rules, it reads, I know it's kind of hard to read, reads one strand and synthesizes a new one. So what DNA polymerase is going to do, I'll just write DNAP here. What it's going to do is it's going to read each of these strands. Let me go ahead and erase this. It's going to read each of these strands. And as it does so, it's going to build a new one using base pairing rules. So what it's going to do is it's going to go along and read this strand. And as it does so, it's going to put in the adjacent base. So it just follows simple rules. Wherever it sees an adenine, it puts a thymine. Wherever it sees a thymine, it puts an adenine. Wherever it sees a guanine, it puts a cytosine. Wherever it sees a cytosine, it sees a guanine, so on and so forth. And this DNA polymerase is going to do the exact same thing. And we're going to have the same exact process here. And if you look here, before you know it, we now have two newly synthesized DNA molecules using the semi-conservative model. What happened is that the strands were separated thanks to the enzyme helicase. Then when the strands were separated, the enzyme DNA polymerase 3 read the existing strands and synthesized a new strand using base pairing rules. So if you look here, we now have two DNA molecules from one. And once again, it is called semi-conservative because it is kind of the same. It takes two existing strands, makes two or synthesizes two new strands, and then you have two separate DNA molecules. All right, so we're going to talk about some types of biotechnology. And I'm not going to get into it too much because we can practically have a whole other lecture on it. And we do need to get to some other things, but I'm just going to tell you the basics of it. And I think we might be having a biotech lab for Labster. but we're going to talk about a couple of these. Biotechnology has changed the world because what we've been able to do now is we've been able to take computers and use them to analyze genetics. So biotechnology, in short, combines computers with genetics. Now, when the SARS-2 virus was first identified in Wuhan, what they were able to do is take the virus and sequence its genome, which is what I showed you with the NCBI database, which is amazing. And what's even more amazing is I think Moderna, they actually had a vaccine ready to go like just a couple days later. Because what they could do is that because they had the virus's genome sequenced in a computer, they found just the part that made the spike protein, and they were able to make a vaccine that, and I don't want to get into it too much, but basically this vaccine um, uh, has the exact code as the SARS-2 virus's spike protein, and it tells your cells, hey, make this protein to these exact specifications. So your cells make the spike protein, and then the immune system responds appropriately. So in the future, these vaccines called mRNA vaccines, they are going to change how vaccines are created because you don't have to use heat-killed viruses anymore or rhinovirus vectors. Instead, you can just make a tiny piece of genetic material that tells your cells to make a very specific protein seen on a virus, and then your cells are going to make that very specific protein. You're going to have an immune response, and then if your immune system ever sees that protein on a virus, it's going to already be able to neutralize it. So biotechnology has absolutely changed the world. But there's a couple of technologies here. And the first one is abbreviated PCR, but it's called polymerase chain reaction. And this is very important because what it can do is it can take just one molecule of DNA and it can multiply it millions of times. So it can create copies. And this is amazing technology, by the way. Create copies of DNA millions of times. So what they can do is they can find just a tiny piece of DNA at a crime scene, just extremely small. And what they can do is put it in this machine, and they can make copies of it to the order of thousands to millions of times. That way, you can have plenty of genetic material found from a crime scene to do testing with. But this has many different applications. You can use this copy genetic material for example, laboratory work. When I had my mouse colony, we had to sequence their DNA to look for a very specific mutation. So what we had to do was take a couple of cells from the mouse. We put it in the PCR machine, and we got millions of copies of DNA, which is much easier to do experimentation with. Something else is called a restriction enzyme, and this is really interesting. So restriction enzymes are actually found in bacteria. And what they do is they look for very specific DNA sequences and cuts it in pieces. This is actually a defense mechanism for bacteria. what it does is these enzymes look for very specific sequences that are not really found in bacteria, and it just cuts it, which destroys the genetic material. But this is extremely interesting because restriction enzymes cut everyone's DNA a different number of times, and in different sizes. So, for example, we can have Bob's DNA, we can have Fraser's DNA, and we can have I don't know, Charles' DNA. Charles. And if we add in a restriction enzyme, because we call that it looks for very specific sequences, maybe it will cut Charles' DNA like this. Maybe it will cut Bob's like this. Maybe it will cut Fraser's like this. So what that does is it's going to create different sized fragments. And this is very important because if you find DNA at a crime scene and you want to link it to someone, all you do is you take the DNA at the crime scene and add in the restriction enzymes, and then you take the suspect and then you add in the restriction enzyme, and if the DNA at the crime scene has the same cuts and sizes as the suspect, then you found your criminal. And this also is, and this is just keeping it fairly short, this also makes up the DNA fingerprint. So DNA fingerprint is the number and sizes of fragments made from restriction enzymes. And this will never change. Of course, there are different restriction enzymes. The most common one is called EcoR1, but the idea behind it is that because your DNA doesn't change, a certain restriction enzyme will cut your DNA in the exact same number of pieces in the exact same sizes each and every time. So Bob's DNA will always be cut like this. It doesn't matter. You can do it 10,000 times. But if you add the same restriction enzyme to Bob's DNA, it will cut it like this every single time. All right, so now we're going to talk about mutations. And talking about mutations is going to be kind of hard without talking about transcription translation. But basically, DNA mutations, what's going on here is basically random changes in genetic code changes DNA. It also changes the shape of the protein. So we're going to talk about this probably I think it's next lecture. I haven't read ahead in your textbook. So how the order is DNA contains information for making proteins. Then you have another piece of genetic material called mRNA, and don't worry too much about this because you'll get the big picture soon. So mRNA brings the instructions for one gene to the rough VR. So what this mRNA does is it scans the DNA code, copies just one piece of it that contains a gene, and then takes that gene to the rough VR. And then on the rough VR, ribosomes read codons and create protein. Now, what is a codon? We are going to review this again. Don't worry. I'm just trying to give you the background so you understand what's going on. So DNA is arranged like this. It's just as a long sequence of nucleotides, but the way the ribosome reads them is it reads them in threes. And every 3 nucleotides we call a codon. And what the ribosome does is, depending on the codon, it puts a very specific structure called an amino acid. So there's 20 odd amino acids, and different codons correspond to different amino acids. So I'm just making this up at this point, but maybe this is arginine, maybe this is valine. And the order of the amino acids determines the protein function. I guess you can say, think of DNA as a recipe book that contains every recipe known to man and woman. The mRNA just copies one recipe from this book, and the ribosomes on the rough VR, they're like the bakers that read the recipe and add the ingredients, which is the amino acids, and then it creates the final product, which is the protein. So once again, if you like my analogy, DNA is a gigantic recipe book that contains every recipe known to man and woman. And the mRNA is just a copy of one recipe in this giant book. The ribosomes on the rough VR, they are the chefs. And as they read the recipe, they add the ingredients, which is like the amino acids. And then when all the ingredients are added, you have the final product, which in our real life example is a protein. So codons specify for different amino acids. Okay, now a mutation is when something happens with one of these nucleotides here, and the result can be variable. Now, the first type of mutation we're going to talk about is called a substitution. And a substitution is when one nucleotide is substituted for another. Now, the effects here are variable. So one nucleotide is substituted for another. So this is going to be our sample sequence. C A T C A T C A T. Cat, cat, cat. Now, how this is going to correspond to the amino acid sequence is cat is read by the ribosomes, and it tells it to put in a protein called histine. His, hiss, hiss. Get it? Cat hiss, because cats hiss. Like, you couldn't have planned this better, right? Now, a substitution is one of these nucleotides here are going to be changed. And there are three types of substitutions. The first is called a silent mutation. And in this case, one nucleotide is changed, but amino acid code does not change. Amino acid code remains the same. And I'll show you an example here. So this is a codon table. You'll be getting very used to this soon. So I'm going to show you a codon table. You can use any one you want to. I'll show you my favorite one. I like this one because it's nice and color-coded. Now, if you look here, you're going to see something kind of interesting. Ignore the use for now because, yeah, we haven't talked about it yet. Actually, let me show you a different codon table that is not. Gosh, they aren't there. Yeah, just pretend for now that the U's are T's. Yeah, you'll see why there's U's here instead of T's when we get to our next lesson. But if we see here CAU, which in our case is CAT, codes for histidine. Excuse me, I said histine before. We also see that CAC also codes for histidine. And if you look at arginine, CGT, CGC, CGA, and CGG all code for arginine. Now, why is this? This is called redundancy, and it is a protective mechanism against mutations. Because mutations can be really bad. And what if there was a way where you can make several different versions or several different codons that are just changed by one letter, several different codons that create the same amino acid? Well, that gives a protective mechanism against mutations. So in a silent mutation, you can have a substitution that just changes one of the amino acids. So it reads CAC, but it's not going to change the amino acid code because CAC and CAT code for the same amino acid. It would be like if there was an error in a recipe, I don't know. Instead of adding Kroger brand flour, instead add in Vaughn's brand flour. I don't know. I don't bake. I microwave. So even though it's changed, even though the recipe has changed, it doesn't actually affect the final product. So the end result is you are none the wiser because the product is the same. Now the next one is called a missense mutation. And the missense mutation is a bit more insidious. In this case, nucleotide is changed. Nucleotide is changed. and amino acid is changed. As a result, protein is changed. So what happens here is that, let me put up a codon table on here. Codon table. I should have one as the background of my phone whenever I get to genetics, since I'm teaching genetics to everyone right now. I should just go ahead and have one on as the background on my phone. In fact, that might be a good idea for you all. Your friends will be very jealous. You're like, oh, cool backgrounds. They have pictures of like their significant others, their kids, their dogs, and you're like codon table. All right, now what if CAT changes to CAA? If that happens, histine is going to change to glycine, abbreviated GLN. Now what's going to happen is that our protein code is going to go from his to glim his. Now what are the effects? The effects are variable. By variable, it just depends on, it just depends. It's kind of like if you were to substitute, for example, a chocolate chip cookie recipe, instead of normal sugar, you add in sugar in the raw, yeah, it's going to change the recipe, but not a ton. But what if you add, instead of sugar, what if you add like salt? Yeah, it's going to change it a lot. So missense mutations can create anything from a non-functional protein to a partially functioning protein. An example of a missense mutation can be seen in sickle cell anemia. The shape of hemoglobin changes slightly, but it still retains partial functionality. It doesn't function as well, but it still retains partial functionality. So an example is going to be sickle cell anemia. It just is called by a single missense substitution. Let me see, and this is impromptu, so forgive me if I don't find a perfect image. But let's go ahead really quick and let me see if I can find you an example. All right, so sickle cell anemia missense mutation. Let's see what I can find here. Because the protein is always changed if the amino acid code is changed, even if it's just slightly. Yeah, so if we see here, if we are going to compare the DNA sequence for hemoglobin between normal hemoglobin and the sickle cell mutation, we see here that there is just a small substitution that changes that just basically changes the orders of the T's and the A's. You'll see why, but this is going to change the RNA code, and it's going to change just one amino acid from glutamine to valine. So what effect does that have on the protein? Well, let's look here. Well, we see here that it's actually going to cause the hemoglobin to change shape, and it's going to cause the red blood cells to change shape, just because of one single nucleotide substitution. And yes, these red blood cells, they can still function, but it's going to open up more complications, such as sickle cells can clot a lot easier. Let me see here. I wish they'd show the protein structure so I can kind of show you how it looks, but I'll have to search quite a bit for that one. But yeah, that just gives you an idea of just changing one nucleotide, changing one amino acid can cause some pretty drastic differences. All right, so Now we're back here. And there's another kind of substitution called a nonsense mutation. So one thing that you will learn about is that there is a type of amino acid called a stop codon. So what a nonsense mutation is it causes a premature stop codon. And what this does is it causes the protein to be shortened, called truncated. Now, let's go ahead and say that this is going to be our DNA code now. So we're going to say that it's going to be cat, 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 cat. And I'll just say like TAA, cat, cat, whatever. So this is going to give us his, 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 his. TAA is going to be actually UAA was the worst one I could have done because that's also stop. Okay, I'll just do I'll do UAC. So TAC, which is going to be tyrosine and then his. So what a nonsense mutation will do is it is going to change just one nucleotide, but the change that it does is going to change it from a normal amino acid to a stop codon, an early stop codon. And because of that, you see here that this protein did not have a chance to finish. It would be like if a chocolate chip cookie recipe says like add flour, add sugar, add eggs, then it just says stop. You'd be like, okay, if that's the recipe, okay. So a stop codon is needed because whenever a protein is being made, the stop codon says, okay, this protein is fully made, stop making it and let it form. But a nonsense mutation is where there's a single substitution that causes just one nucleotide to change, creating an early stop codon, creating a truncated or shortened protein. Now, the effect is also variable. And usually how it works is that the earlier, well, actually, let me put it this way. The more premature the stop codon, the worse the effect. Usually, any kind of nonsense mutation will create a non-functional protein. But in theory, the earlier the stop codon is, the worse the effects. Like, let's say it's at the very end of the protein sequence, like we see here. The protein could have partial functionality. Like, what if the only part that was cut out was put cinnamon on top of the chocolate chip cookies? I don't know. You're still going to have mostly the finished recipe, mostly the finished product. But if it stops after add flour and sugar, you're not going to have anything close to a chocolate chip cookie. All right, so these are substitutions, and next are the really nasty ones, and we call these frame shift mutations. And they're called frame shifts because they change the entire reading frame. And there's two types of frame shift mutations. We have additions and insertion and deletions. So an insertion is where a nucleotide is added, and a deletion is where a nucleotide is removed. But as you'll see, the effect is actually the same. So a deletion is the nucleotide is changed, is removed. All right, now we're back to our original sequence: cat, 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 cat. Now with an insertion, what's going to happen is that a nucleotide is going to be randomly inserted. So I'll just say a G. Because of that, it's going to change the reading frame by changing the codons, because our new sequence is going to look like this. CAT, which is going to stay the same. Then we have GC. Then we have GCA, TCA, TCAT. So look at how much has changed. We've gone from his, 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 to his. And what's GCA? His, alla, alla. Now TCA is going to be, let's find it here. UCA is going to be serline. So sir, and who knows what this is. So more or less what the insertion did is it completely changed the amino acids after the point of the insertion. Now, deletion has very similar effects. Cat, 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 cat. Instead, what's going to happen is one nucleotide is going to be removed, just excised, and it's going to change to CAT, ATC, ATC, AT. So we're going to go from his to his. ILE, ILE, and who knows what this is. So once again, the entire reading frame is going to be changed. So what frameshift mutations do is that they change the codons. So codons are altered due to addition or deletion of a nucleotide. So what it does is it basically messes everything up. Now, what is the effect? Bad, to put it nicely. Usually, frameshift mutations create a completely dysfunctional protein. But in theory, if the frameshift mutation is at the very end of the sequence, it could retain partial functionality. Very similar, the earlier in the sequence the frameshift is, the more dramatic the effects. If the frameshift is at the very end of the sequence and not many subsequent codons are affected, then it could retain partial functionality. I know that a lot of this is not going to make a ton of sense right now, but after you learn about transcription and translation, it's going to make a lot more sense. So I do suggest you go back after we learn about transcription translation and either look at the PowerPoint or rewatch this part of the lecture, because it might make more sense depending on how much you currently know about genetics. All right, everyone. So that's all the content for this week. I know that this week was very dense, but genetics is a incredibly important component of biology, especially with how much biotechnology is progressing. And if you find this stuff interesting, then I highly suggest that you give this some thought as far as a possible area of focus when you transfer to your 4-year university. But yeah, fascinating stuff. Hope you guys enjoyed learning it, and I'll see you guys soon. Over and out.